The topic that I'm going to tell you today is about the bio-convergence system of layered double hydroxide nanoclay that I have studied so far. As you can see the slide, a number of papers on functional nanomaterials covered with nanoparticle, nanocomposite, nanopore has exponentially increased over the period of last 2 decades. And it was found that their application fields are mainly focused on biomedical, healthcare science, energy, and environment applications. As you can see here, it is expected that more than 70,000 papers will be published in the near future in the field of nanomaterials. The first thing, and first in general, advanced nanomaterials can be categorized into 3 different types of nanostructure: nanoparticle, nanopore, and nanohybrid or composite with multiple functions. Those functions of clay-based material have enabled me to study on biomedical science and new nano-bio-info-cognos and energy environment applications such as carbon dioxide capture. Let's think about what the blueprint is then for those nanomaterial engineering regarding clay science. I think that there are two big trends and driving forces based on each national research direction. For example, since National Science Foundation in US reported the importance of convergence of nano-bio-info-cogno in 2002, many scientists have tried to combine nanotechnology with other bio-info-cogno technology for creating new global market. On the other hand, as you can see the right-hand side, the Paris Agreement on climate change has resulted in acceleration of sustainable technology such as solar cell, fuel, a secondary battery, something like that, for reducing greenhouse gas and utilization. Based on those background, let me show you my first research topic on nanomedicine. As you know, there have been many reports on biomedical science using various nanoparticles, including cationic polymer, liposome, mesopro-silica, and carbon nanotube, etc. Among them, I'm very interested in layered double hydroxide nanoclay, which is applicable to targeted drug delivery system, molecular imaging, and biosensor detection. Let me show you what the LDH clay is and the reason why I'm interested. The LDH is one of 2-dimensional inorganic material, and its chemical formula can be written as divalent and trivalent metal ion with interlayer anion. Crystal structure is started from brucite clay mineral, magnesium hydroxide, and also similar with that of hydroxide clay in nature. The LDH clay has positive charge within the edge shared octahedral layer by isomorphic substitution of divalent metal ion with trivalent metal ion, and then charge balancing anionic gas molecule such as carbonate could be occupied between metal hydroxide interlayers. Those occupied anions can be replaced by exchangeable other molecules, including DNA and anti-cancer drug molecules. The global pharmaceutical company has supplied the hydroxide as name of tarsid for the use of regulation and neutralization of strong acidic condition inside the stomach, which means that LDH with composition of magnesium aluminum is quite biocompatible due to the fact that LDH is soluble in acidic environment, but it is insoluble in neutral and basic conditions, so that LDH is very useful as a bio drug delivery carrier with controlled release property. In my research, intercalation chemistry has been used for engineering bio-inorganic hybrid clay materials. As you can see here, reversible insertion of biogas species into the host LDH lattice with layer structure would be achieved by driving force such as mainly electrostatic interaction and van der Waals hydrogen bonding interaction at the surface interface between the biomolecular and two-dimensional inorganic hydroxide layer. In such a way, LDH nanoclay can have therapeutic function for gene, chemo, photo, and combination therapy, as you can see in this slide. Our research team has reported many research and review articles on how LDH nanoclay work and behave inside animal body and cancer cell when the LDH nanoclay are injected into the body. The key point that I'd like to stress on here is that the LDH nanoclay with controlled particle size of 100 nanometer and high colloidal stability could be prepared by hard thermal reaction at the temperature of 100 Celsius. Also, it would be accumulated into the tumor site by enhanced permeation and retention reaction, and then easily internalized inside cancer cell through the clathrin protein-mediated cellular uptake mechanism, which have been well proven by in vitro cell line experiment by my supervisor, Professor Gino Choi. However, there was still no clear evidence at the level of in vivo animal experiment at the time. So my attempts have been made to demonstrate the passive and active targeting of LDH clay nanoparticle by siRNA delivery and cancer therapy. As you can see here, active nanoparticle with 100 nanometer was prepared by conjugation reaction with cancer cell specific ligand, such as folic acid, onto the surface of LDH nanoparticles through the silicon coupling reaction, as reported by Professor Gemino. In my study, therapeutic siRNA was loaded by self-assembly reaction to specifically knock down gene called survivin gene in cancer. By doing so, active LDH-FA nanoclay is expected to be delivered into tumor tissue by exploiting both the EPR effect and ligand receptor interaction. As you can see here, ex vivo biodistribution study on KB tumor-bearing nude mice clearly confirmed that active LDH-FA nanoclay was preferentially accumulated into the tumor within 30 minutes after IP injection, which is 1.2-fold higher than only EPR effective dependent passive type of LDH nanoclay. From animal experiment by collaboration, the mice group treated with active targeting function of LDH-FA nanoclay was shown to be more effective in suppressing tumor growth. As expected, we achieved the significant inhibition rate of tumor growth rate, as well as suppression of specific gene product with around threefold higher level than those in the LDH with passive targeting. Furthermore, according to survival curve, the mice group treated with LDH-FA siRNA showed around 80% survival after 30 days post-treatment. On the other hand, the mice treated with PBS control group were all dead after 30 days post-treatment. In order to prove the biocompatibility, we investigated the change in body weight and histological examination of mice after IP injection with high dosage for 5 weeks. As shown here, no significant toxicity was observed, even though systemic delivery of LDH-FA resulted in substantial accumulation in both the liver and spleen by non-specific RS uptake. Since we discovered cellular uptake mechanism of LDH nanoclay, we also found that the LDH nanoparticle with 100 nanometer size show the direct cytosis pathway, which can enhance the early endosomal escape in order to minimize damage and degradation of therapeutic gene molecule by releasing them into the cytosol rather than late endosome and lysosome. Finally, we conclude that the LDH nanoclay particle with 100 nanometer size are very useful for gene delivery system. Let me move to the second topic of nano-bio-info-cogno system. These slides show you different type of synthetic method for the declaration of gas species into interlayer nanospace of LDH nanoclay, from coarse precipitation, conventional ion exchange to exploration and reassembling method, which is recently proposed and intensively used to incorporate large size and bulky molecules. In our previous study, it was very successful in preparing layer by layer type of DNA LDH hybrid material, which was intercalation of double helix DNA molecule into interlayer space of LDH by conventional ion exchange method. However, the intercalation method was not able to protect DNA molecule with a long chain due to the presence of this closed unintercalated dangling part of DNA, as you can see here. In order to provide the breakthrough, I tried to prepare a co-share nanostructure type of DNA LDH hybrid by exploration reassembly method, as you can see here. Through the cross-section TM image, we observed the artificial DNA molecules with a core with a core size of 100 nanometer are encapsulated within the nanospace made by LDH nanoshell with a spherical morphology. Furthermore, we demonstrate the LDH nanoshell can play a crucial role as a reservoir of DNA molecule containing information of the base sequence. In the thermal stability test, the melting temperature of artificial DNA inside LDH was 27 Celsius higher than that of free DNA with 35 base pairing. In the case of chemical stability, the encapsulated DNA was protected from the copper catalyzed oxidative radical stress. Also, the bulky artificial DNA was also completely protected from the biological enzyme attack condition, which was confirmed by electroporating mobility and biochip detection analysis. Based on those results of bio-clay material, our research group suggested the concept of NBIC conversion system along with proof of principle experiment. First thing was, as you can see the left side, was a nano-forensics system with microarray biosensor detection technology. And second strategy was Arbata DNA code system with camera recognition technology of smartphone. In order to demonstrate nano-forensic system, the hybrid material was mixed with black color ink as authentic information. If needed, the hybrid material can be collected from the signature, and then DNA molecule could be DNA molecule is able to be released out of nanospace of the LH clay with treatment of acidic solution, of which base sequence information can be detected and read out with a DNA chip biosensor assay by customized design, as you can see here, where each spot was fabricated with complementary DNA sequence to detect pre-assigned DNA security. In a second strategy, the detection image of microchip was fabricated as a symbol image of QR code or color code, so that smartphone camera installed with decoding application software can recognize those biochip image as QR code or color code. And then DNA information can be decoded on the smartphone screen from this database web server through wireless remote connection. Those are what I proposed for Arbata DNA convergence with clay hybrid material. This slide shows two examples of item level tagging system for the feasibility demonstration. As you can see, the left side, we developed systemic origin traceability barcode system of agriculture product by demonstrating a blind test to discriminate the origin of mixed ginseng item. We purchased three kinds of ginseng from different origin, alpha, beta, and X. The ginseng from origin X was used to negative control. In the encoding step, two kinds of barcode DNA were prepared with different base sequence information. One is A prime for origin alpha, and the other is B prime for origin beta. The DNA chip also was fabricated at the same time to detect each barcode DNA molecule if needed later. If DNA chip matched with the each barcode DNA molecule, it will display alphabet L shape, green color, red color on the different corner, something like that. For encrypting step, each DNA barcode molecule was encapsulated by LH clay, and each DNA LH hybrid solution was sprayed on the ginseng surface of head part. And then in decrypting step, acidic treatment with EDTA followed by recovery was carried out to collect each origin barcode DNA. Last decoding step was done by DNA chip. So blinding test could discriminate ginseng origin from alpha and beta correctly. Finally, we demonstrate also Arbata DNA nanohybrid system, as you can see the right-hand side. In the encoding step, DNA clay hybrid material was printed out using conventional inkjet print. In such a way, stabilized DNA molecular tag can be labeled onto the surface of product packaging label. In the critical step, after scanning the tags under fluorescence illumination, we were able to discriminate the real DNA tag by detecting them with a smartphone. In such a way, we were successful in demonstrating bio-convergence system with hybrid engineering technology based on clay material and science. Thank you so much for your kind attention. Let me finalize my presentation.
Oh. Okay, thanks, Professor Park. Now we have 3 minutes to ask questions. Any questions?
Okay. Professor Park. Hello?
Hello, yes.
Yeah, we can hear you.
Yes, please.
Park, can you hear me?
Yeah, I can hear you.
Okay, I just you know, have a question about, you know, sometimes the particles for the control of the LDH for injection of this kind of medicine, does it will it block the blood vessel or not?
In my knowledge, usually from 80 nanometer to 100 nanometer cannot block the blood vessel of animal body.
So is there any side effect if this kind of solid particles injected into the blood vessels? For example, even the decomposition or give the alumina and magnesium is have toxicity in the body.
If the surface of LH nanoclay is too high, for example, LH particle with 50 nanometer. In the case of LH clay with 50 nanometer, the clay has higher surface than that of LH nanoclay with 100 nanometer, which lead to higher toxicity because of higher surface area.